Is the unit ops project keeping you up late? Is regular coffee just not doing it anymore?
Then caffeine extraction might be right for you. I'm Jason Hall. You might remember me from class. And today, our cutting edge team is going to take you behind the scenes and explore step by step how to extract your own caffeine.
Extraction is a separations process that utilizes the difference of a molecule solubility between an aqueous phase and an organic phase. For a successful extraction to occur, the molecule being separated must be much more soluble in one phase than the other. Due to the two phases immiscibility, the target molecule and its respective solvent can be easily collected as there are two distinct liquid phases as seen in the figure below. For this experiment, caffeine is the target molecule and the organic solvent is dichloromethane. The source of caffeine will come from ground coffee beans. The grinds will be placed in a reactor filled with a steady state flow of water. The outlet water will be collected and then used as the aqueous solution for extraction with dichloromethane. After performing the extraction, the solvent and caffeine mixture will be placed in a spectrophotometer, where absorbance at 275 nanometer light will be measured. Using a calibration curve, these readings will be converted into the concentration of caffeine in the solvent. The purpose of this experiment was to investigate the effect of several factors of this process on the concentration of caffeine extracted. In our experiment, we examined the following: the mass of coffee placed into the reactor, the size of the coffee grinds being placed into the reactor, and the flow rate of water into the reactor.
As always, make sure you're wearing the proper protective equipment, lab coat, gloves, and don't forget those goggles, bud.
Coffee beans are the source of caffeine for this experiment. As such, the beans are poured into the coffee grinder, which is then set to the selected grind level. Once the top has been secured, the grinder is started and the beans are grounded up. Here we can see the grounds on the coarse setting, the medium setting, and fine setting. The coffee grinds are secured in the reactor using a normal tea bag. First, the bag is carefully opened and the tea leaves emptied out. Then, the selected mass of coffee is placed in the bag and stapled shut. Next, the reactor setup is prepared. Approximately 2 liters of water are added to the feed tank and the temperature set to 80 degrees Celsius. Before any experiments can be performed, the flow rate must be calibrated. The flow rate meter is set to a specific value and water is collected for 30 seconds. The collected water is then weighed to calculate the flow rate. The process was repeated 5 times in order to create a calibration curve. Now the reactor can be set up. The volume of the reactor is marked off by tape in order to provide a consistent reference point throughout the different trials. Once a flow rate is chosen, the reactor valve must be adjusted in order to keep the water level at the taped line. The tea bag can then be placed in the reactor so it is just resting beneath the surface of the water. After allowing the coffee to sit in the reactor for 2 minutes, sample collection can begin. A funnel is placed in a small flask which is set below the reactor. Once the sample level has risen to the marked 15 milliliter line, the next sample may be collected. A total of 3 samples are taken per experiment. After all 3 samples are taken, extraction can begin.
First, 15 mils of dichloromethane is measured out in a graduated cylinder. A pipette is used to add or subtract small amounts to ensure accuracy. Next, the dichloromethane is added to the extracted caffeine solution present in the separatory funnel. Now, the stopper is placed in the funnel. The separatory funnel is removed from the clamp and inverted the desired number of times, in this case, 5. Extra care is taken to ensure the stopper does not come loose. The funnel is placed back in the clamp and the stopper is removed. Note how the 2 phases of liquid have separated from each other. The funnel is slowly opened to drain out the dichloromethane with the extracted caffeine. This process is repeated for each of the 3 samples taken from all trials. A small amount of the final solution is added to a quartz cuvette, which is then placed in the spectrophotometer. The absorbance is read and recorded at 275 nanometers.
When finished with the experiment, all waste must be disposed of in the appropriate waste containers. Then wash out the reactor with DI water. The following graph shows the extracted caffeine concentration as a function of the mass of coffee placed into the reactor. As can be seen, our results show the trend that as the mass of coffee placed in the reactor increases, so does the concentration of extracted caffeine. This can be explained by the fact that increased mass of coffee in the reactor leads to more caffeine available to be transferred to the water. Since volume in the reactor was held constant, this would lead to a higher concentration in the exiting aqueous solution. This next graph shows the effect of coffee ground size on the extraction concentration. Our data shows that as the coffee particles move from fine to coarse, concentration of extracted caffeine increases. A possible explanation is that the finely grounded particles were more tightly packed together in the tea bag, resulting in less water being able to percolate through the entire grounds. Ultimately, this reduced the effective surface area of the mass. With a coarser grind, it can be theorized that water was able to come in contact with more of the coffee, resulting in more collected caffeine. Finally, the effect of flow rate on concentration was also examined. The results show the trend that as flow rate is increased, concentration decreases. This makes sense because volume is kept constant, so by increasing flow rate, residence time is decreased in the reactor, which leads to less time for caffeine to be absorbed by the water.
Well, there you have it. That's how you do caffeine extraction. To learn more, call the number on your screen. Catch you on the flip.